Hello, welcome to this webinar on human milk oligosaccharides, the frontier of infant nutrition and health. The webinar is sponsored by Biostime Institute for Nutrition and Care. My name is Dr. Ardythe Morrow. I am Professor of Pediatrics, Epidemiology, and Nutrition at the University of Cincinnati College of Medicine and Cincinnati Children's Hospital Medical Center. I've spent a career studying human milk oligosaccharides, or HMOs, actually for about 20 years. Research on HMOs is now causing a lot of excitement and is being undertaken in universities and laboratories worldwide, bringing new understanding to the field of HMOs on an ongoing basis. HMOs are truly a frontier of infant nutrition and science, and I wanted to try to convey my wonderment as a human milk researcher. What better way to do that than by invoking the wonders of the Milky Way, the galactic frontier for our little planet Earth. So I've used the Milky Way as the backdrop on this slide and throughout my talk. And by that, I mean to convey the great adventure that human milk research is. Before embarking on our scientific journey about HMOs, I wanted to set the stage by telling you the story that is pictured here in a beautiful painting by Peter Paul Rubens entitled The Birth of the Milky Way. The painting is based on an ancient Greek myth. In this myth, Zeus, the father of gods, who's pictured on the left, and with a crown on his head, brings a child to his wife, the goddess Hera. Hera begins to nurse the child and then suddenly realizes that the child belongs to her rival, the rival for Zeus's affections. Hera then pulls the child off of her breast, which causes her milk to spurt in an arc across the heavens. In this myth, the stars of the night sky are imagined to be the precious drops of the goddess's milk. Galaxy then refers to the arc of milk, or the Milky Way that we see in the night sky. Galactose is a Greek word referring to milk that we now use in science to refer to a major milk sugar that you'll hear about again. So mention of the Milky Way makes a fitting start to discussion of the importance of discovery and understanding of human milk oligosaccharides. In the journey of this talk, let's begin at the beginning, the importance of breastfeeding and mother's milk. Then I'm going to define and provide an overview of HMOs, their structures, their composition in human milk, the functions, and the known and potential health effects. So we begin with mother's milk in the grand background of the Milky Way. Breastfeeding is the goal and the gold standard of infant nutrition. Exclusive breastfeeding for 6 months should be followed by continued breastfeeding with complementary feeding, and this is the global public health and nutrition recommendation, which is clearly and deeply evidenced. As health professionals, we must do all that we can to support breastfeeding. Broadly speaking, breastfeeding improves infant outcomes in 3 major ways pictured here: healthy immune function, healthy growth, and improved or enhanced cognitive ability. In this next slide, I cite a much more detailed list of conditions for which there is specific clinical evidence of benefit from breastfeeding, specifically the ways in which breastfeeding protects infant health and supports healthy development. Review of this list, though, makes evident that breastfeeding has broad impact on lowering a variety of infectious diseases, such as severe lower respiratory tract infections, acute gastroenteritis, acute otitis media, and reducing risk of a broad range of immune disorders, including inflammatory, allergic, and autoimmune conditions. In other words, breastfed infants have a generally healthier immune system and better development. It's also well established that breastfed infants have significantly greater cognitive development. This is known not only from epidemiologic studies, but also trials. And finally, the World Health Organization has clearly demonstrated that breastfed infants have a significantly different growth than typically found in formula-fed infants. In other words, there remains a real gap in health effects comparing formula-fed to breastfed infants. Breastfeeding is also a major influence on development of the early infant microbiome, and this has been made abundantly clear in a series of studies over the last 5 to 10 years. This slide conveys the finding of a very major study called the TEDdy study, multi-site study of thousands of infants from multiple countries. The TEDdy study examined the formation of the microbiome over the first 4 years of the child's life. During the first 12 months of life, breastfeeding is the dominant influence on the formation of the infant microbiome, and it particularly contributes to the early dominance of bifidobacteria. After addition of solid foods to the infant diet, there is a rapid increase in microbial community diversity and a reduced abundance of bifidobacteria. Here in this slide, I'm sharing my own microbiome data from the Cincinnati site of a study known as Global Exploration of Human Milk, which was sponsored by Mead Johnson. The results of this study echo that of the TEDdy study. We had fecal samples from 89 predominantly breastfed infants who were analyzed at the times shown in this slide at 4, 13, 26, 52, and 104 weeks. This is relative abundance of specific microbiota. The dark blue or purple at the very bottom of each bar represents bifidobacteria. And you can see that. It was shown in the TEDdy study, previous slide, that bifidobacteria predominate in exclusively predominantly breastfed infants up to 6 months of life or 26 weeks. And then as solid foods are introduced, the bifidobacteria abundance declines, microbial diversity increases in response to this major dietary change. But how is it then, through all of these various health effects, that breastfeeding can have such broad effects on infant health and development? For my money, human milk is influencing the development of the microbiome and the reduction of health disease conditions through its unique biomedical composition or through bioactive components. For example, human milk contains oligosaccharides, glycoproteins and glycolipids, secretory antibodies, cells, cytokines, chemokines, defense proteins, and so on and so on. But of all of these compounds, the most abundant and arguably the most multifunctional protective component of human milk may be the HMO fraction. So let's go on to understanding what are HMOs. And in the next few slides, I'll provide an overview. The photo in this slide is one of my favorite because it's a vial of mother's milk taken from a freezer in my lab, where I have actually thousands and thousands of vials of mother's milk for analysis. And if we were to extract all of the water found in mother's milk, we would be left with the solid components only. This graphic overlays the vial of mother's milk to indicate the relative composition of the solid milk components. So lactose is the most abundant, followed by the lipid fraction of human milk. And then after the lipid fractions, the human milk oligosaccharide fraction is the most abundant. Similar to HMO in abundance is the quantity of protein in mother's milk. Now think about that HMO is at least as abundant or is more abundant than protein in mother's milk. And that fact alone is enough to suggest the importance of HMOs in infant nutrition and why it's so important to study. So again, what are HMOs? As I mentioned, HMOs are the most common solid component of human milk after lactose and lipid. There are more than 200 different HMOs that have been identified by glycomic analysis, but by definition, they are sugars of 3 to 32 individual monomers in length. And they are based on lactose to make more complex structures. These structures are unique to human milk and include homologs to gut oligosaccharide structures, making them really uniquely human. To provide a more specific understanding of HMO structures, I'm providing this map to the HMO galaxy set against, again, the Milky Way. Beginning with a key on the upper right corner, there are only 5 individual sugars or monomers that provide the building blocks to make the HMOs. Each has been given its own color and shape. Glucose is pictured as the blue circle. Galactose, remembering that word from our earlier slides, galactose is the yellow circle. And the amide derivative of glucose is pictured as a blue square. Then there's fucose, the red triangle, and sialic acid, which is the purple diamond. These symbolic representations of individual sugars have become the universal picture language of glycobiology. So you may well see these same symbols again in other talks. 2 of these individual sugars, glucose and galactose, blue circle and yellow circle, link to become the disaccharide known as lactose, shown on the left. This is the most common sugar of milk and the backbone for all of the human milk oligosaccharides. Here are some of the neutral human milk oligosaccharides that are considered precursor molecules, called precursors because they can be added on to making longer and longer chains. Another key component of mother's milk oligosaccharide is the monosaccharide fucose, a very important individual sugar, red triangle, which is found in more than half of all individual HMOs. And here we see 2 trisaccharides, some of the simplest and most abundant HMOs, but very important, 2-fucosyllactose, or 2FL, and 3-fucosyllactose, 3FL, in which fucose is linked to lactose in different ways. There are other more complex molecules that include fucose, as is shown here. And then here is the molecule known as sialic acid, an acidic oligosaccharide that can be linked to others through specific alpha 2,3 and 2,6 linkages. And here are the 2 most abundant and simplest of the sialylated oligosaccharides, and again, the sialyl oligosaccharides can be much more complex and large as well. So sialyl oligosaccharides comprise a very important fraction of HMOs, particularly in earlier lactation, perhaps 20 to 30% of total HMOs. Altogether, there are about 15 to 20 HMOs that comprise the vast majority of the HMO fraction in all mother's milk. But there are also many other HMOs found in mother's milk in very small quantities that can vary from mother to mother, and perhaps as many as 200, but extremely small quantity. Here I'd like to compare for you what the structure is of HMOs compared to other prebiotics or oligosaccharides that are sometimes in use in infant formula. At the top is the HMO that I've studied the most, and that's 2-fucosyllactose, that simple trisaccharide sugar. To the right are some of the more diversities of the different human milk oligosaccharides. But you can see that it is quite different visually from the next oligosaccharide, which is commonly used, which is fructo oligosaccharide, or FOS, F-O-S. This is made up of a chain of fructose individual sugars, which are not naturally found in mother's milk at all. And at the very bottom then, there is another oligosaccharide that is commonly used now in infant formula and other things, which is called GOS, or galacto oligosaccharide. And you can see that GOS has yellow and blue circles, that is to say glucose and galactose, and it's a chain of those sugars. But it also does not contain either fucose or sialic acid, and so in many ways it is quite different than the human milk oligosaccharides that we're talking about. Do these structures matter? Human milk researchers think that it does matter. Its functions generally differ based on differences in structure, and I'm going to be telling you about some of the many functions that HMOs have that often are not found with either FOS or GOS. For example, as I show in this slide, the oligosaccharide 2FL on the right, it is structurally similar to the mother's or the infant's own endogenous mucosal oligosaccharides, for example, found in the gut. 2FL on the right has the yellow and blue circles connected to that red triangle, and on the left, a yellow and blue square connected to the red triangle. This structural similarity is showing that fucose is at the terminal end linked to galactose, and that this commonality of structure makes them similar in terms of being able to bind to specific kinds of receptors and other functionalities. Again, different than FOS or GOS. So now I'd like to address the question, what is the HMO composition of mother's milk specifically? And I'll address that question in the next few slides. But before we start on that section, I'd like to ask you to place your bet. Do you think that mother's milk oligosaccharide composition is highly standardized, pretty much the same from mother to mother? Or do you believe that it's probably highly variable? So with that question, let's look at the data. Here I compare the HMO composition that my group measured in milk samples from mothers at 4 weeks after delivery as part of the GEHM study, comparing in the upper pie chart here, samples that were collected in Shanghai, China, to the lower pie samples collected in Cincinnati, Ohio, 2 very different populations. Each individual HMO is represented by a different color. The black and gray at the top are the precursor oligosaccharides. The blue, those made by the with alpha-1,2 fucose linkage, the green alpha-3,4 glucose linkage. Whoops, and I skipped ahead here. I'm going to need to get back to where I was. Excuse me for a moment. You can see ahead what we're going to be talking about. Okay, here we are. That's slipping my finger. So what I was going to say is that looking at the color distribution, you can see that the HMO profiles, we've got the same colors around the circle. It's also apparent that the profiles differ somewhat in the relative size of each slice of the pie. So, for example, the precursor oligosaccharide LNT, represented in black at the top, is somewhat bigger in the Cincinnati milk samples than perhaps in the Shanghai milk samples, and so on. As you go around the pie, there can be relative differences in abundance of specific oligosaccharides. But overall, the general HMO pattern seems to me more similar than it does seem different from population to population. Overall, regardless of population, about 70% of the oligosaccharide is neutral and about 30% is sialylated, but the precise amounts differ. Now here is a slide representing an important study called the childbirth cohort that was conducted in Canada under the leadership of Megan Azad. She was wanting to address the question, what influences HMO composition? First, she found the total HMO content of mother's milk, even within one country, varied fourfold. Second, she found that the individual HMOs that we were talking about could vary as much as 20-fold from mother to mother. Though there was a specific interest in looking at the effect of diet, there was no association between HMO and diet quality, an important question that mothers often ask. Rather, there was an association with fixed characteristics that the mother cannot influence, and that included secretor genetics, stage of lactation, and the geography, season, parity, and ethnicity issues. So I want to address something that may be new to you and seem a bit strange, because I use the phrase secretor genetics for a moment. And so let's review what that actually means. Secretor genetics, we're referring to a gene that's known as the fucosyltransferase 2 or FUT2 gene, and that has major polymorphisms or genetic variations in the gene that can be different then from mother to mother. And this strongly influences the oligosaccharide profile found in individuals' mother's milk, also in other secretions like saliva, and in the gut, the infant mucosa, and even in the lung. Remember the slide in which I introduced the idea that similar oligosaccharide structures are found in mother's milk and the maternal or infant gut. And here again, we picture 2FL in milk on the right and the H antigen made by the same gene in saliva and gut pictured on the left. So both of these structures are made by the same single enzyme, which is the product of the FUT2 gene known as the secretor gene. So here we have a genetic influence in a way on infant nutrition. In this slide, I'm showing a visual representation in a bar chart of the relative HMO composition among Cincinnati mothers in relation to maternal FUT2 secretor status, depicting the most common individual HMOs or groups of HMOs. This graph represents relative abundance, so each oligosaccharide or group is represented as a percentage of the total. The left-hand bar represents the genetically positive FUT2 positive mothers known as secretors, and they are about 3/4 of all mothers. Secretor mothers have a full set of oligosaccharides, and these mothers, 40 to 50% of HMOs, include an alpha-1,2 fucose linkage, is what that enzyme makes. And these are represented in the graph in shades of blue. So you see 2FL, LNFP1, LNDFH1, and LGFT in blue. Other fucose-linked oligosaccharides are made, and these are shown in green and yellow shades. And after that, the precursor molecules in shades of brown, then the acidic oligosaccharides represented in black. Now, in contrast is the bar on the right, which represents the milk of about 1/4 of mothers known as non-secretors. And these non-secretor mothers don't have any part of that bar that is blue, because they do not make an entire class of HMOs with the alpha-1,2 fucose linkage. Instead, non-secretor mothers produce more of other oligosaccharides. You see that there is more green and more yellow. These are other types of fucose-linked oligosaccharides, 3 or 4 fucose linkages. And they make more acidic oligosaccharide in black. Another important point, though, is that quantitatively, overall, non-secretor mothers have on average about 30% lower HMO content overall of their mother's milk. This difference may not be important in many circumstances, but evidence is growing that there could be advantages and disadvantages to being secretor or non-secretor in different circumstances. And that's why humanity has a variation in this capability. And research over the next decade, I believe, will help to clarify what those advantages and disadvantages are. This slide here represents a global picture of HMO composition of mother's milk in a study of 11 global sites in Africa, Europe, the US, and Peru. The study was directed by the McGuires, who live in Idaho in the United States. It was conducted in collaboration with Dr. Lars Boda and colleagues at each site. This provides a global view in terms of the representation of the prevalence of secretor human milk among these populations. And you can see all women on the left is 100%, but when they're divided up between non-secretors and secretors, they're about 23% non-secretors and 77% secretors, which is about what I would expect for a global average. And what they're showing here is the total oligosaccharide, and you can see that it's lower in the non-secretors than in the secretors, and you can also see that there's variation from site to site, again for reasons that we have yet to fully understand. We've found so far from Megan Azad's work and others that geography, and McGuire said geography influences mother's milk oligosaccharide, and that genetics, we've shown, influence oligosaccharide in milk. But here is represented in this slide a randomized controlled trial of an important missing piece, which is maternal microbiota. This study by Seppo et al. Is of a probiotic trial that was conducted in Northern Europe, and reports that maternal microbiota can influence HMO composition. This study specifically reports on the composition of the milk of 81 women randomized to receive either probiotic or control. The intervention group received a probiotic mix, whereas the controls did not. And you could see that there are significant differences between probiotic or placebo in 2 major human milk oligosaccharides. And overall, the probiotic fed mothers had an increase in specific types of oligosaccharide, type 2 and alpha-2,3 oligosaccharides, and decrease in alpha-2,6. Now, the health consequences of this difference, no one knows. This particular study, however, teaches us something important, that maternal microbial colonization or microbial influences can also influence mother's milk composition, at least to an extent. So this is an important piece of the puzzle. But it's not enough to know all of these things that I just described about HMOs. Well, what do we know? That it's uniquely tailored to human babies, it's structurally diverse, so that there are various influences that make mother's milk oligosaccharides somewhat different from mother to mother. But with that background, we now need to move on to HMO functions. That is, address the question, what do HMOs actually do and why should we care? So broadly speaking, there are 3 major HMO functions as indicated here. First, HMOs help manage the infant's microbiota. Second, HMOs contribute to optimizing host defense against harms to the infant of various kinds. And third, HMOs appear to enhance neurodevelopment. And I'll review each of these topics, providing a few examples under each. Before we do that, I would like to discuss here for a moment a concept of the gut-immune-brain connection. To link to the previous slide, the idea that HMOs may influence each one of these domains might seem a far stretch, but science over the past 10 years or so has, in its own other domains of work, demonstrated that there is a connection between gut, brain, and immune systems. And let's consider. First, the immune system is made up of many different structures and processes, but about 70% or so of the immune system is found in the intestinal tract, which is the largest immune organ in the body. Gut-associated lymphoid tissue, known as GALT, is made up of many cell types and cytokines representing both the innate and adaptive immune systems. And the gut also contains microbiota, which produce metabolic products that can enter circulation, and a large enteric nervous system, which can signal to the brain via the vagal nerve. So the gut, immune, and nervous systems are really all very, very interconnected in a network of bidirectional communication. In the following slides, I'll discuss how HMOs might influence this triad. So let's begin with perhaps one of the most well-evidenced pieces about the idea that HMO support to the growth of beneficial bacteria. What happens to HMO when the baby consumes it? Well, it traverses the small intestine and largely makes it through the small intestine to the colon. A small quantity is absorbed and found in circulation. But once the vast majority of HMO makes it to the colon, it becomes support for the growth of beneficial microbiota and the production from those bacteria of beneficial metabolites. Represented in this slide is a small snapshot of the findings of the Newberg lab, which shows the important strains of bifidobacteria and Bacteroides which reside in the colon, and that they can ferment a set of different oligosaccharides listed at the top of the table here, including several fucosylated and sialic acid containing HMOs. Then the metabolites of bacterial fermentation produce short-chain fatty acids that benefit infant gut health and immune function. But HMOs do more than nourish these individual beneficial bacteria. In this review paper represented here, Andrea Markobal summarized the research of several different human microbiome research groups who found that provision of HMO nourishes a beneficial microbial community overall, pictured at the top cartoon image. Carbohydrate substrate from HMOs supports an entire beneficial community, as shown in the gut lumen in that cartoon. The middle cartoon image depicts the circumstance of infants deprived of HMOs. And if the infants receive no other form of prebiotic carbohydrate in their diet, the microbiota use the infant's own gut mucus layer for nutritional support. That is, they burrow down into the mucus layer and utilize it in order to stabilize the microbial community. When this leads to an adhesive community of bacteria that dine on and embed in the outer loose layer of mucus. Now, finally, in the lower cartoon, depicts what can happen during times of gut disturbance if there has not been human milk oligosaccharide or other prebiotic carbohydrate in the diet. Pathogenic bacteria can become embedded in that mucus layer, and then they can use the gut disturbance to get to the surface of the intestinal tract, attach, and cause infection. And given the evidence for this concept, a diet rich in oligosaccharide or other prebiotic carbohydrates appears very important for maintaining a healthy microbiota and the health of infants, and indeed people at all ages. There are other ways that HMOs function to help infants manage their gut microbiota. And besides the prebiotic effects I described before this, human milk oligosaccharides, because of their unique structure, also can act to bind pathogens and inhibit infections in another way. And in this slide, I describe the concept. Many enteric pathogens, including noroviruses, invasive campylobacter, rotaviruses, Vibrio cholera, strains of diarrhea genicans, E. coli, and so forth, have been shown to bind to the structure in the gut known as H. antigen, also made by the secretor enzyme, which you remember from previous slides. Now, this cartoon on the right shows an incoming pathogenic E. coli, which has a tropism for fucose receptors, and it has something called an adhesin that protrudes and can bind to the oligosaccharide receptor on the surface of the infant epithelial cell. And the pathogen binding to the epithelial cell is the first step that leads to infection. But if there is human milk oligosaccharide in the gut, during times of pathogen presence, the pathogenic species can use this to bind, and this is now considered a decoy receptor, the human milk oligosaccharide, and the binding then to this human milk oligosaccharide molecule can flush the pathogen out of the system through peristalsis. Now, in the next slide, I want to show you scientific evidence that HMO2FL reduces the carriage of invasive campylobacter, so is support for the previous concept. This is a figure from a paper published by my colleague, Dr. Guillermo Ruiz-Palacios, who's in Mexico City, in the journal JBC. His laboratory showed that invasive strains of campylobacter bind to H. antigen in the gut and also to 2FL in milk. And they tested the ability of 2FL to reduce the carriage of invasive campylobacter. So they challenged a series of mice with invasive campylobacter. Then they gave half 2FL and half they just gave their controlled diet. And then you can see that those given 2FL had much lower log levels of campylobacter in their stool, in the red line there, much lower than in the controls. So this provides support for the idea that there can be a receptor decoy function in regard to the specific structural binding of a given pathogen. But does this HMO concept of binding work similarly in human infants? Well, here I show the first epidemiologic study of this concept, which I conducted in Mexico and published in 2004 on mother's milk and risk of infant diarrhea. We studied 93 mother-infant pairs, measured HMO levels in the milk of mothers, and then grouped the mothers as having low, intermediate, or high levels of 2FL, as shown in the graph there on the bar chart on the left. And so in the mothers with low 2FL levels, the left-hand bar, the risk of Campylobacter diarrhea in their infants was 9 cases per 100 child months, and much higher than the 2 or so episodes per 100 infant months if the mother's milk had intermediate or high levels of 2FL in milk. And that we also found when we looked at 2FL and other related secretors, HMOs in milk, a similar pattern was seen in reducing the risk of lower, moderate to severe diarrhea of all causes in infants. So since this is also the same pattern as seen in our bench studies, we found this to be important evidence of potential public health impact of HMOs. Now, in the next slides, I'd like to address the second major function of HMOs, and that is optimizing host defense. And optimization of host defense is cleverly done in a variety of different ways, and many of the HMOs individually have specific roles. So this is a table published in a review paper by another colleagues, Triantis, Boda, and Van Nierven. And the table is a complex summary, but it's essentially saying that HMOs can bind to many different receptors throughout the immune system. So the immune system has a lot of different cell types and a lot of different receptors, and HMOs individually in different structures are matched up to those receptors. And it indicates the systemic potential for different HMOs to influence immune status. For example, a set of fucosylated oligosaccharides, including 2FL and 3FL, bind to DC sign and influence immune response to antigen presentation. Other neutral oligosaccharides bind to galectins on T cells and other antigen-presenting cells, lymphocytes, and so forth. And sialylated oligosaccharides bind to Siglecs found on other immune cell types like neutrophils and monocytes and dendritic cells. Selectins influence leukocyte adhesion. So in other words, there are multiple ways in the immune relevant cells in which human milk oligosaccharides have and can have an influence. So in the previous slide, I gave a broad overview, and in this slide, I want to just take one specific experiment that just demonstrates immunomodulation by HMO on gut cell response. Specifically, again, in the Newberg lab, they found that the HMO known as 2FL, lowers inflammatory response of gut tissue exposed to pathogenic E. coli. So in this first part of the graph, you see the inflammatory expression of gut cells measured in terms of the inflammatory cytokine TNF-alpha. And as would be expected, at baseline, just looking at baseline tissue expression, TNF-alpha expression is low. But then when the gut tissue is exposed to pathogenic E. coli, TNF-alpha expression spikes. When 2FL is then added to the gut tissue, TNF-alpha levels return to low pre-baseline levels, even though the tissue was still exposed to the pathogen. So this and many other such experiments demonstrate tissue-specific immune adaptation to HMO presence. A very different strategy for HMOs optimizing host defense is by improving the response and repair function, and that is gut adaptation and repair following gut injury. And a number of animal studies have been done, performed in different labs, of the HMO 2FL. And just in brief, summarized here, this one oligosaccharide can restore intestinal perfusion after gut injury by upregulating nitric oxide synthase, eNOS, and promoting vasodilation. That's the study by Good et al. It can enable growth and biogenesis after gut injury. That's a study by Mezov and Weiss et al. And I'll be talking about the Mezov study. And it can also reduce the severity of necrotizing enterocolitis, not probably NEC itself, but the severity thereof. There's several studies that have been shown that. So let's talk about one of those studies. The example of HMO improving host defense after injury is a study done at Cincinnati Children's by our team, led by Ethan Mezov and Mike Helmrath, a very talented pediatric surgeon at Cincinnati Children's. And he does an animal model of called ileocecal resection, which creates a short gut. So you've got the injury in this case is a surgical intervention. So we wanted to see if 2FL could improve growth and restore the gut architecture, and it did. It also increased beneficial microbes and increased the bioenergetics of gut cells. Now I'm going to show you this next slide. It's showing something that's called transcriptomics. It's looking at the expression of genes on gut cells. And when we found that in half of the half of the mice who were given the surgery and given 2FL, that the expression of the small bowel genes of different genes was significantly different than those who are not given 2FL after surgery. And what did 2FL associate with? Well, the genes that increased had to do with energy processing. So you had a much more energetic gut cells. They're also producing more. Their biosynthesis processes were upregulated. And also, once again, we saw upregulation of genes having to do with host defense or host microbial interactions. Again, supporting previous studies that I've shown you. And then finally, I'd like to get to the final of the three approaches that the HMOs do to benefit the infant, and that is neurodevelopment. And this is one of the most important domains of infancy, and it's a hot area of investigation, but it's still a relatively new domain of research. So in this slide, I'm going to do, again, a conceptual overview. How can HMO, which is found in the gut after being consumed, possibly influence the brain? Well, it is shown in here, the blood-brain barrier is intact and does not allow the entry of HMOs into the brain. But HMOs can infect the brain by affecting the gut microbiota, and this influence can alter signaling from the bacteria to the vagal nerve, which is a vast system in the gut, which can in turn signal to the central nervous system, and that's the middle system. But also, HMOs can alter the metabolites produced by gut microbiota, specifically the neurotransmitters, for example, GABA, histamine, serotonin, other metabolites that have the potential to influence brain health after absorption into circulation. So that's the concept. I'm going to just show you one of the several preclinical studies that's been done by different groups indicating that HMO administration can influence neurobehavior, particularly cognition or learning, anxiety or stress, and sociality. These are the domains which have been identified so far. And this one study done by a talented group in Spain, Vasquez et al., shows that chronic administration of 2FL increases the efficiency of operant conditioning, or I'll call it learning, in rats. Specifically, rats were rewarded with a food pellet for pushing a lever, as shown in panels A and B. And this followed a very specific protocol. And there were 4 groups of rats compared. First group, and shown in the red bars, 2FL fed groups, and in controls. And then there were, in each one of those 2 groups, half were given what's called a bilateral vagotomy, so the vagal nerve was removed to prevent signaling from the gut. And the other group just remained intact and to induce the same level of stress that surgery would do. It was a sham surgery. So in brief, in the 2FL fed rats who are not vagotomized, again, the red group in the red bar, the rats learned faster to perform the desired task. And this experiment not only supports the finding that oligosaccharide can influence neurodevelopment, but it also gives us a specific mechanism, which is vagal nerve signaling. This also relates back to my previous slides showing that breastfed infants have generally higher cognitive development, and gives us a mechanism, one of several mechanisms by which this may be occurring. So there I've shown you so far a lot. We've covered a great deal of territory, but the studies I've shown so far have been preclinical, largely at the bench. There are really only so far a few trials of HMOs in infants, and those trials are actually quite important. What are the health effects of HMOs in infants? I would like to now share the results of 2 major infant feeding trials that tested HMO in the context of infant formula. So first, I'll begin with the randomized controlled trial conducted by Abbott investigators looking at growth, tolerance, and immune response to HMO, specifically 2FL fortified formula. The study enrolled 420 infants in a randomized controlled trial. There were 3 arms with formula, and all of the formula arms had oligosaccharide at the same concentration, which is 2.4 grams per liter. The control formula contained solely GOS. The intervention arm, first one, tested 2FL at 1 gram per liter, and then GOS at the remaining concentration of 1.4 grams per liter. And the next intervention arm at a different dose of 2FL, very low, 0.2 grams per liter, and GOS at 2.2 grams per liter. And then there was a 4th study group, which were exclusively breastfed infants. Each of these groups had about 400 infants each. So the primary readout of the trial, again, was tolerance, growth, and immune modulation. The trial found the formulas to be well tolerated, all of them, and no significant differences in growth by study group. Now, in this slide, I'm going to show you a little bit of the immune modulation data from that same trial. So blood samples were collected from studied children, and here I show specifically that only the data from TNF alpha levels, because we've talked about TNF alpha before as an inflammatory cytokine. And the control group is shown in the green bar on the left, and the study groups of infant feds with formula at 2 different dose levels are shown in the purple and pink bars, so that you can see the TNF alpha levels in the control formula group was significantly higher than those in the formula-fed groups with 2FL. And this experimental data in infants supports the findings of some of the preclinical studies that I reviewed with you. But TNF alpha was not the only difference in peripheral cytokines. Indeed, there were 10 cytokines evaluated, of which 5 were different between the formula and control groups. And the yellow I should mention that yellow bar across represents the exclusively breastfed infants, so you can see what range, and you can see that when 2FL was given, it was in the same range of cytokine expression as in the formula groups fed 2FL. So there is one more randomized controlled trial that I'd like to review with you, and this trial was conducted by Puccio and others, published in 2017, it was sponsored by Nestle. And they studied 175 infants, and the test group had formula with added 2FL and LN-NEOT, which is a precursor oligosaccharide neutral. And so the total quantity of oligosaccharide was 1.5 grams per liter. Over the 12 months of follow-up of this trial, test versus control infants had significantly lower parental reports of bronchitis. It's test group 10% versus control infants 28%. Lower respiratory tract infection was significantly lower, 19% versus 34% in controls. And they had significantly lower antibiotic use, which is a major finding of public health significance, 42% lower in the test infants versus 61% in the control infants. So these are very different outcomes as each tested a different set of conditions, but both studies are consistent with the concept of HMO impact on immune function as we've previously described. So I've really told you a lot here, and it's a lot to absorb. We've gone pretty far distance. So let's review the HMO journey that we've been on together. So I've presented data indicating that promotion and support of breastfeeding is a critical public health priority, and that HMOs appear to contribute to the health impact of breastfeeding. I've shown that HMOs are structurally distinct and yet vary between mothers and populations, but the HMOs also are structurally similar to our own endogenous oligosaccharides in our gut. Shown that preclinical studies indicate multiple functions, that HMOs help manage the infant microbiota, they promote host defense, enhance neurodevelopment. And while clinical studies, that is randomized controlled trials, are yet few, the results are very supportive of this overall concept, demonstrating tolerance and significant immune benefits. So we continue as human milk researchers to expand the frontier of infant nutrition science through research. I just wanted to show you how fast the research is now going. With the discovery of individual HMO molecules was by Georgi and Kuhn in the mid-1950s. And in the 70-some years since that time, you can see over the decades, there was a steady climb, and this scale is the number of publications on HMO in peer-reviewed journals that has been steadily climbing. But even the last, say, 10 years, there's been an almost exponential increase. And over time, we've been learning about the variety of HMOs, about their prebiotic and pathogen binding effects, about their effects on host immunity, and most recently now, excitingly, also on neurobehavior. And now we have more than 125 publications on HMOs per year from scientists all over the world. So I'm excited to continue on this frontier of discovery. And now, after this long journey, we're coming back to Earth. And I'd like to thank you for joining me on this journey about HMOs. So if you have any questions regarding this webinar, the BINC team invites you to send them a message until the 5th of January 2020. You can contact BINC, the email listed on this slide. My name is Ardythe Morrow. I'm at the University of Cincinnati, and I thank you for your time and listening to this webinar. Thank you.